Okay, so welcome back to Chapter 45, Part 2, which we're going to be talking about muscle contraction. So this is going to be a little bit involved, so I will be posting as well some additional helpful animations for you guys to check out, so make sure you do those and use them to help you when you're answering the mastering bio and the discussion questions if you need to. So this is a topic that I spend a lot of time on with my anatomy students when I taught high school. I think I spent a month on it, and when I teach anatomy physiology, it's generally a more complex topic for students to understand. So make sure you're kind of focusing in on this. All right, so the mechanism for how muscles work is something that's been looked at for a long time, and before microscopes we really didn't know how it worked. But it all comes down to this structure here called the sarcomere. And we're going to break this down in a couple of slides coming up. So when we look at muscle tissue, specifically skeletal and cardiac, we're going to be focusing on skeletal. It's made up of those muscle fibers, long, slender muscle cells. Okay, so inside of those muscle fibers, there's even smaller fibers called myofibrils. So we've got, think about it, our whole muscle. Our muscle is packaged up into these muscle fibers, and now we're going to pull out from these muscle fibers myofibrils. If we look on the myofibrils, there's these striped patterns due to light and dark alternating bands, and those are called sarcomeres, which are on the myofibrils, which are in the muscle fibers, which are the cells that make up muscle tissue. Okay, when a myofibril contracts, the sarcomere becomes shorter and smaller, and when the muscle relaxes, the sarcomere gets longer. So you can kind of imagine it like in your mind, a slinky kind of, and all of the pieces kind of overlapping there, all of the coils overlapping is sort of how it works. So again, let's kind of orient ourselves with where we are. So here's your muscle. Your muscle tissue is made up of these bundles of muscle fibers. Okay, it's actually called a fascicle. And you can pull out a muscle fiber. So this is one cell. And then if you look at that one cell, we've got a myofibril, and along that myofibril, we've got tons of repeating sarcomeres. So here's a sarcomere, here's a sarcomere. And what's going to happen is that sarcomere is going to, all of those sarcomeres, all along the myofibril, are going to contract when the muscle gets shorter. When the muscle gets shorter, it pulls on the bone and causes movement. So it's all about getting shorter to cause movement. So how does this work? So it's the sliding filament theory is how it works. So there's two types of filaments that are in the sarcomere. So there's thin filaments that are made up of actin, and there's thick filaments that are made up of myosin. So these filaments are going to overlap over each other, okay, in certain parts. So the myosin is going to overlap, but the light banding patterns are going to stay the same. They're not going to overlap. Okay, and like I said, when you look at the animations I post, it'll make sense. It's kind of hard to show this on a still figure, but like I said, definitely the animations, make sure you watch those. So this sliding filament model involves the filaments sliding past each other and the sarcomere changing in length. So the actual filaments themselves don't change their length, they just cross over each other and shorten the sarcomere. So here's a sarcomere, okay, in between Z discs. That's one sarcomere. So what's going to happen is these Z discs are going to pull towards the middle, towards that H zone, and when they do that, when they pull towards the H zone, okay, you have to imagine this one piece getting shorter, okay, those thin filaments sliding over top of the thick filaments, right, and we see the shortening of the sarcomere. So how does this happen? It's a four-step process, so it has to do with the hydrolyzation of ATP. So on the myosin, these red blobs, there's these little things coming off of it. Okay, those are called myosin heads. And what's going to happen is these myosin heads are going to hook on to the actin. And when they hook onto the actin, they're going to pull it in towards the center. Okay, so those red bands are not moving. It's just the thin actin bands that are moving because the red myosin bands are pulling them towards the center. So how does this happen? So ATP, which is, the molecule of energy, has to bind to the myosin head, causing a conformational change, okay, that's going to release the head from the actin. All right. When ATP is hydrolyzed, it's going to turn into ADP because a phosphate comes off, and there's the inorganic phosphate, and the myosin head straightens and the head pivots. Okay, so the neck straightens and the head pivots. Myosin head then binds to a new actin subunit farther down the thin filament. So it's like it's going to click on, click off, hydrolyze, click on, pulling this thing towards the center. Okay? The myosin head is now cocked and ready for the power stroke. Okay? When the inorganic phosphate is released, the neck bends back to the original conformation. Okay? And that stroke is going to move the entire thin filament relative to the thick filament. So ATP is released, finally, and the myosin filament is ready for binding to another ATP molecule. So as this process continues of ATP binding, hydrolysis, okay, we see the actin getting pulled in towards the center. Okay, so in terms of what's happening here, like I said, it's difficult to visualize from a still picture, so I really think it's important that you guys spend some time on the animations that I will have posted. Okay, but here's another look at how this is happening. So starts with the myosin head bound to the actin subunit on the thin filament. The myosin ATP binds to the myosin head and it releases it. Okay, so this part is actually really interesting, that the ATP binding releases the myosin head. So really when the ATP binds, it signals the release of the actin and the relaxation of the muscle. So we need energy to relax muscles, which is kind of counterintuitive, right? But if you think about it, when we have someone dies, right, they end up in rigor mortis, where the muscles stay contracted, right, and they never relax because there's no energy to release those myosin heads. When ATP is hydrolyzed, however, we see the head cocked, okay, and now we're starting to move that filament. Step 3, phosphate's released, head pivots. That's the power stroke. Okay, ADP is released, and the cycle ultimately will repeat here. So if someone doesn't have ATP, right, look at what ATP is doing. They're going to be stuck right here. Okay, and that's how we get stuck in rigor mortis. So if anybody's thinking about pre-med, that is a question that always ends up on the MCAT in some way, shape, or form. The MCAT is not something that would just ask you straight-up questions. It would ask you it in a very convoluted way, really testing your understanding. But this is something that comes up a lot. So how do these two proteins stay kind of away from each other or stuck to each other? It has to do with two proteins called troponin and tropomyosin. So when the muscle is relaxed, tropomyosin is covering up the binding sites on actin. And specifically, the binding site that's on the actin is made up of something called troponin. So in order to get this muscle to contract, calcium has to bind to troponin, and the tropomyosin complex moves off and allows for contraction to begin. Again, this is something that the animation you'll be able to see pretty easily, but troponin is on the actin molecules, and it's covered up by tropomyosin when the muscle is relaxed. When the tropomyosin moves off, the troponin binding sites become visible to the myosin to bind to it, and the muscle can contract. Okay, so these are called regulatory proteins because they're regulating this whole system. So there's your myosin binding sites, which are made of troponin, and notice they're blocked by tropomyosin here. Okay, tropomyosin covers them. When calcium comes in, it's going to bind to those calcium binding sites that are actually on the tropomyosin, and now what happens is the myosin binding sites become available to the myosin heads to bind to them and contract the muscle. So pretty cool. So when the muscle is relaxed, not contracting, tropomyosin and troponin work together to block the myosin binding sites. They're totally blocked. Contraction begins when calcium ions are released from the sarcoplasmic reticulum inside of a muscle cell. We'll see how that happens in a second. And now the myosin heads can bind and pull that muscle. Okay, so all of that is happening at the filament level to contract the muscle. What's happening with the nervous system here? So this is the part that we kind of already know from last chapter about action potentials. So in order to contract a muscle, motor neurons need to send an action potential down to the muscle cell and release acetylcholine, which is the neurotransmitter that's going to be sent from the neuron to the muscle cell. So acetylcholine is going to leave the neuron and go into, the synaptic space there, synaptic cleft, and bind to the muscle cell, and then that is going to trigger an action potential in the muscle cell, which is going to signal the release of calcium, which will then go towards all of the things that we talked about in the previous slide. Okay, binding to the tropin and binding sites, tropomyosin moving off, all of that stuff. Okay. So once the acetylcholine binds to the muscle cell, it's going to trigger an action potential, and that action potential is going to move through the cell through structures called T-tubules, which essentially just move the action potential through the muscle cell until it interacts with something called the sarcoplasmic reticulum, which is going to allow for calcium to just kind of explode out of it and go to the filaments to ultimately get them to slide. Okay. So we call this the neuromuscular junction because it's the interaction of the nervous system and muscle tissue regulating muscle contraction and complex movement. Okay, so what does this look like? So here it is. So the first step, and this should look familiar from last week a little bit. Here's our motor neuron, and an action potential is being sent from the nervous system down to the end of the motor neuron to release acetylcholine. Acetylcholine is released into the synaptic cleft until it binds to receptors on the muscle cell. And once those acetylcholine receptors receive the acetylcholine, it triggers action potentials to move all through the muscle cell, all down these T-tubules. And once the action potential reaches the sarcoplasmic reticulum, which is really all throughout the muscle cell, it's going to release calcium. And calcium is then going to go to expose to the tropin and binding sites and allow the tropomyosin to move off and the muscle to contract. Okay, so here's this again, another good picture for you to study going through all of this. Okay, kind of what I just described. Okay. All right, so that wraps up this chapter. Make sure you take the time to watch the animations and complete the Mastering Biology review for this week.